All right, welcome back. In terms of gene regulation, there's one more topic I want us to cover in a little more detail. And that's the process of alternative splicing. So with alternative splicing, this is going to be a way that the cell can generate multiple protein products from one gene. So we can get multiple different proteins that might have slightly different functions from a single gene. Oops, single, there we go. So what I have up here with exon 1, exon 2, exon 3, exon 4, this is going to be our gene. Because with eukaryotic genes, the way they're organized, you're going to have areas that are exons, and then you're going to have intervening regions that are introns. And a lot of times, those introns are the regions of RNA that actually give rise to those microRNAs that we talked about in another video. So those introns sometimes do have some function that may not necessarily be related to the gene that is being transcribed. Sometimes it may, okay? So what we've got here, all right, just so we've got our levels and what we're talking about straight, what I've got in pink here is going to be our DNA. What's going to happen? That RNA polymerase, those activator proteins, those transcription factors, RNA polymerase, are going to come in and they're going to transcribe our gene, okay? And the way that's going to happen is we'll get a transcript with exon 1, exon 2, I made exon 3 smaller just so that it'd be a little bit different. And exon 4. Okay? So this, and maybe what I need to do is make these wiggly. Because now what we're talking about is we have messenger RNA. It's the messenger RNA that can be alternatively spliced. Okay? It might be in terms of the protein that needs to be made. We can have an option where exon 1, exon 2, exon 3, and exon 4 are all spliced together. So the way that process of splicing is going to happen is you're going to have the 3' end of one exon spliced to the 5' end of the next exon, okay? So if we go like this, what we're going to get out of it is a protein that has exons 1, 2, 3, and 4, all right? Where the alternative splice comes in, we might have another option where we get exon 1, exon 3, exon 4. So in this case, our splicing pattern would go something like this. Okay? That's what it means to be an alternative splice product. Okay? And the protein that's made here is going to be different than the protein here because we no longer have exon 2. All right? So that's going to result in a different protein, potentially different amino acids being inserted after exon 1. Okay? Depending on how the reading frame goes with translation. Okay? So all our alternative splicing the alternative splice product is still an RNA product. From this point then, we would get translation, and we'll call this, we're going to translate this to protein 1, okay? Our alternative splice product is also going to undergo the process of translation. So it's going to take that RNA information and convert it into protein, but it's going to give us a different protein. And we'll call that protein 2, just generically, all right? So that process of alternative splicing is a way that the number of genes in the human genome can kind of be maximized, because you can make some different proteins using some of the same exons out of an RNA transcript, okay? Now I only have 4 here. You may have more than that when we're talking eukaryotic cells. You can have 20 or 30 exons that make up a particular gene, and you can see how you would get a lot more diversity in the number of proteins that can be generated out of that, okay? So hopefully that makes sense. If you have any questions, please let me know.